Hi, this is Mrs. Wyrick with Seminole County Virtual School, and I just wanted to give you a quick explanation of the cell division lab assignment for 3.01 cell division. So for this assignment, you're actually going to complete a virtual lab activity, and on our homepage, I do have a template that's already in Word format that you can download of the lab report. But essentially, it's going to look like this page where you've got your title, objective, hypothesis that you have to fill in. And then you are going to complete the data table as you actually go through the virtual lab activity. Don't forget to give an explanation of what's happening in each of these stages, so tell what's going on with the cell. And then you're going to create a graph. I highly recommend a pie chart for your data analysis section. You want to try and determine what percent of the cell's life is spent in each of these phases. You have a conclusion to write, a little conclusion paragraph, and then some analysis questions at the very end. So that's overall what you're filling in. The instructions page gives you a little bit of background information about onion root tips, and we're going to be looking at different cells. So you'll notice here, this is a cell in interphase. Here we have a cell in prophase where the chromosomes are just becoming visible. In metaphase, your chromosomes are lined up on the midline. Anaphase, they start to pull apart. In telophase, they have pulled into two opposite directions, and we see our nuclear membrane starting to reform. And last, in cytokinesis, in plant cells, we have a cell plate, which is a little baby cell wall that starts to form between our two new cells. So when you get into the virtual lab activity, click anywhere to continue. And you're going to be looking through this microscope, and you are going to determine, this would be a great place to get the information for that data table for the record. You're going to determine and classify each of the cells that you see as one of these phases. So for example, when I look at this cell, I noticed that I can't see any of the chromosomes. So that's going to be interphase. And so I just click on interphase and it gets labeled. And then I'm going to go ahead and go to the next button. So this one, I see that my chromosomes have formed. They're not lined up on the midline though. So this is going to be prophase. And then you're just going to continue doing this until you get all the way through your 50 cells, and then you'll be able to see your data markers at the end. You will then repeat this experiment a second time. So this one's going to be interphase. Okay, so you're going to repeat this experiment at the end, or at this a second time, and that is where your second set of data is going to come from. I hope this quick video helped. Good luck, and let me know if you have any other questions. Thanks. Bye.